Welcome to G Fun Facts Online, where we dive deep into the biggest questions in science. Today, we're tackling a fundamental biological puzzle. You, me, every human, we have 46 chromosomes in most of our cells. A dog has 78, a cat has 38. But then you look at a species of fern, the adder's tongue, and it has over 260. On the flip side, the male of an Australian bulldog ant gets by with just a single chromosome. This raises a huge question. If chromosomes carry the blueprint for life, why isn't there a clear link between how many an organism has and how complex it is? Why does a simple fern have 27 times more chromosomes than a human, while a complex social ant has the bare minimum? This is the great chromosome count conundrum, and the answer reveals the messy, surprising, and truly dynamic nature of evolution. Let's get into it. First, a quick refresher. What exactly are chromosomes? Think of them as the ultimate storage solution. If you unraveled the DNA in one of your cells, it would stretch out to be about 2 meters long. To fit all of that into a microscopic nucleus, the DNA is wrapped tightly around proteins called histones, like thread around a spool. This whole complex is then coiled and condensed into the compact, X-shaped structures we recognize as chromosomes. This packaging is crucial for more than just saving space. It helps control which genes are turned on or off, and it ensures that when a cell divides, each new cell gets a perfect copy of the genetic library. In most animals, including us, we are diploid, meaning we get 2 complete sets of chromosomes, one from each parent. These form homologous pairs, one from mom, one from dad, carrying the same genes, though maybe different versions of them. Humans have 22 of these pairs, plus 1 pair of sex chromosomes, XX for females and XY for males, making 46 total. This complete set of chromosomes, its number and appearance, is called a karyotype, and it's a signature for a species. For a long time, the intuitive assumption was that more complex organisms must have more genetic material and, by extension, more chromosomes. It just makes sense, right? Building a human must require more instructions than building a yeast cell. The amount of DNA in one set of chromosomes is called the C-value. And the shocking lack of a relationship between this C-value and an organism's complexity is famously known as the C-value paradox. The paradox is impossible to ignore when you look at the numbers. A Japanese flower called Paris japonica has a genome 50 times larger than a human's. Some single-celled amoebas have genomes up to 100 times larger than ours. And as we mentioned, the adder's tongue fern, with its 260+ chromosomes, completely dwarfs our human count of 46. It works the other way too. The muntjac deer, a fairly complex mammal, has as few as 6 chromosomes in females. So it's crystal clear. The number of chromosomes, or even the total amount of DNA, is not a reliable measure of complexity. The solution to this paradox started to appear in the 1970s with a groundbreaking discovery. Most of our DNA doesn't actually code for proteins. In humans, less than 2% of our genome consists of protein-coding genes. The rest, once controversially dismissed as junk DNA, is anything but. We now know this non-coding DNA is a vast and vital collection of sequences. It includes introns, which are segments snipped out of gene transcripts before a protein is made. It includes powerful regulatory elements that act as switches, telling genes when and where to turn on and off. And it includes massive tracts of repetitive DNA, with sequences repeated over and over, as well as jumping genes called transposable elements that can move around the genome, and ancient broken copies of genes called pseudogenes. The sheer amount of this non-coding DNA varies wildly between species, and this is the main reason for the C-value paradox. An organism like that fern can have a huge genome and tons of chromosomes, simply because it's accumulated a lot of this non-coding material over its evolutionary history. Chromosomes are just the shelves in the genetic library. What really matters is the content of the books, the genes, and the intricate system for how those books are used, not the number of shelves they're organized on. This brings us to the core of the issue. How do these numbers change in the first place? The diversity we see today is the result of a dynamic evolutionary shuffle, where mistakes during cell division can, over millions of years, create entirely new karyotypes. Let's look at the mechanisms. One of the most dramatic ways to change the count is chromosome fusion, when two chromosomes break and rejoin to form a single, larger one. The most famous example of this is in our own family tree. Humans have 46 chromosomes. Our closest living relatives, chimpanzees, gorillas, and orangutans, all have 48. For years, this was a puzzle, but genetic analysis has found the smoking gun. Human chromosome 2, one of our largest chromosomes, is the result of a head-to-tail fusion of two smaller chromosomes that have remained separate in all the other great apes. The evidence is undeniable. A normal chromosome has one centromere, which is the pinched-in part in the middle, and protective caps called telomeres at both ends. Human chromosome 2 has the remnants of a second, inactive centromere, right where you'd expect it to be if two chromosomes had joined together. Even more tellingly, it has telomere sequences buried in its middle, which makes no sense unless they were once the ends of two separate chromosomes. This fusion event happened in our ancestors after our lineage split from the chimpanzee lineage, and it became a defining feature of what would become Homo sapiens. We have even seen this happen in the lab. Recently, scientists successfully fused chromosomes in mice, creating a new, stable karyotype, proving that such a major evolutionary leap is entirely possible. The opposite of fusion is fission, where one chromosome splits into two. This increases the chromosome number. For this to work, both new, smaller chromosomes have to acquire a functioning centromere and telomeres to stay stable during cell division. While fusion might be more common, fission has also played a major role in the evolution of many groups, like butterflies, where larger chromosomes seem to be more prone to splitting, driving rapid changes in their karyotypes. But the most drastic mechanism for increasing chromosome number is polyploidy. This is where an entire set of chromosomes gets duplicated. Instead of being diploid with 2 sets, an organism might become tetraploid with 4 sets, or hexaploid with 6, and so on. Polyploidy is incredibly common in plants. It's estimated that up to 80% of all living plant species are polyploids, and we can see evidence of ancient whole genome duplications in the history of almost all flowering plants. It usually happens when errors during the formation of sperm or egg cells lead to gametes that are diploid instead of haploid. If 2 of these unreduced gametes fuse, a tetraploid individual is born. This is a powerhouse of evolution because it can lead to instant speciation. A new tetraploid plant might be reproductively isolated from its diploid parents. If they try to breed, they produce triploid offspring with 3 sets of chromosomes. These triploids are usually sterile because their chromosomes can't pair up evenly during cell division. This reproductive barrier effectively creates a new species in a single generation. That record-holding adder's tongue fern? It's the product of repeated rounds of polyploidy over millions of years. This isn't just an evolutionary quirk, it's a major source of innovation. The extra sets of genes provide a safety net. One copy of a gene can keep doing its original job, while the other copies are free to mutate and potentially evolve entirely new functions. Polyploid plants also often show gigas effects. They have larger cells, leaves, flowers, and fruits, which can give them an ecological edge. This increased genetic diversity can also make them more adaptable to environmental stresses like drought or disease. While much rarer in animals, polyploidy is not unheard of. It's found in some insects, goldfish, and salmon. And most profoundly, deep in the evolutionary history of all vertebrates, including us, there were at least 2 rounds of whole genome duplication. Many scientists believe this event provided the raw genetic material for the explosion of complexity that led to fish, amphibians, reptiles, birds, and mammals. Finally, there's a 4th mechanism: aneuploidy. Unlike polyploidy, which involves whole sets, aneuploidy is the gain or loss of a single chromosome. This is usually caused by an error called non-disjunction, where chromosomes fail to separate properly during cell division. In humans and most animals, aneuploidy is almost always bad news. The genetic imbalance it creates disrupts the finely tuned network of gene interactions needed for development. We see this in conditions like Down syndrome, which is caused by an extra copy of chromosome 21, or Turner syndrome, a female with only one X chromosome. Most other aneuploidies in humans are so severe they result in miscarriage. But from an evolutionary standpoint, it's not always a dead end. Plants are much more tolerant of it. And even in other organisms, aneuploidy can sometimes provide a strange advantage. In yeast or cancer cells, for example, gaining an extra chromosome can sometimes confer resistance to drugs. Over long evolutionary timescales, the gain or loss of a chromosome, followed by other rearrangements, can contribute to the gradual change of a species' karyotype. These changes in chromosome number aren't just abstract genetic events. They have profound consequences. The most significant is their role in creating new species. As we saw with polyploidy, if a group within a population develops a different chromosome number, they may no longer be able to produce fertile offspring with the original population. This reproductive isolation is the very definition of how new species are born. Even a single fusion event can contribute. A hybrid individual with mismatched chromosomes will have trouble in meiosis, leading to reduced fertility and discouraging gene flow between the groups. Over time, the two groups diverge and become distinct species. We also see a special case of this in the evolution of sex chromosomes. In many species like ours, the X and Y chromosomes started out as a perfectly matched pair of regular chromosomes. But about 300 million years ago, a gene on one of them mutated to become a sex-determining gene. To protect this new maleness gene, the area around it stopped swapping genetic material with its partner chromosome. Over millions of years, this lack of recombination led to the gradual decay and shrinking of the Y chromosome, which is why it's now so much smaller and has so few genes compared to the X. And while large-scale changes drive evolution, aneuploidy often plays out on the level of individual health. In animals, a balanced genome is so critical that the gain or loss of a single chromosome is often devastating. This is why aneuploidy is a leading cause of developmental disorders and pregnancy loss in humans. It's also a hallmark of cancer. Over 90% of solid tumors have aneuploid cells. The relationship is complex. This genomic chaos can both promote tumor growth by allowing cancer cells to rapidly evolve and adapt, but it can also create stresses that work against the cell. It's a double-edged sword that is central to cancer biology. So, in the end, the chromosome number puzzle isn't really a paradox. It's a window into the messy, imperfect, and endlessly creative process of evolution. The simple idea that more chromosomes equals more complexity is wrong. The staggering difference in chromosome numbers across the tree of life is not a ladder of complexity, but a historical record of each lineage's unique evolutionary journey. This journey is shaped by a mix of random chance, a fusion here, a duplication there, and the constant pressure of natural selection. For some, like ferns, accumulating massive numbers of chromosomes through polyploidy was a winning strategy, providing genetic backup and fuel for adaptation. For others, like that Australian ant, a highly streamlined genome with just one chromosome proved just as effective. Ultimately, the number of chromosomes matters far less than the information they carry and the complex rules that govern how that information is used. The fact that a fern can thrive with over 1,000 chromosomes while an ant thrives with one is a powerful testament to the incredible flexibility of life's coding system. It shows us that in evolution, there is no single best way to build an organism, only a magnificent diversity of strategies that have stood the test of time. Thanks for listening to G Fun Facts Online.